All right. So I would like to welcome you to July's seminar, and I have the great pleasure to introduce Professor Giovanni Bottegoni. Professor Bottegoni did his PhD in medicinal chemistry at the University of Bologna. Following his graduation, he moved to the Scripps Research Institute as a research fellow and also followed then by a postdoc. And then he later became also a team lead at the Italian Institute of Technology. It was during this time that he also co-founded the company Biki Technologies. And he then worked at Sosei Heptares before moving to University of Birmingham and now being professor at University of Urbino. His research interests are in the development and application of computational methods for developing polypharmacological drugs, and he also loves Lord of the Rings and playing Dungeons and Dragons. So with further ado, thank you, Giovanni, for having us. And yes, we're very interested in hearing about your research.
Thank you very much, Sonja, for the very kind introduction. I'll proceed right away with sharing my screen. There it is. Let me just go in presentation mode. There it is. So can you confirm that you see it right, full screen and everything? Yes. All right. Yes, perfect. Thanks a lot. So welcome, everyone. Again, thanks for the very nice introduction. We'll talk today about characterization of allosteric pockets in an environment at the interface between protein and membrane. I have several examples, but let me get started with saying that being my approach mainly computationally driven, defining, characterizing these sort of pockets in the specific region basically boils down to combining two different modeling techniques, being docking and molecular dynamics. So what you'll see today are different flavors in the way these two techniques can be combined. And I understand most of you are familiar, at least on a, let's say, theoretical level with these techniques. I understand that many of you are basically using these techniques or have used these techniques at least during their secondments. But I'll provide, at least for docking, a very brief introduction just to make sure that everyone possesses at least the basics of what we are going to talk in a little bit more detail. So this is a brief outline of the presentation. Without further ado, I'd like to get started with talking about molecular interactions. And our current understanding of molecular interaction is traditionally credited to Ehrlich. Basically, he challenged somewhat what was then the main idea when he said that basically corpora non agunt nisi fixata, which means that molecules do not interact unless they are bound. This was a direct challenge to the idea which pre-existed, which was that corpora non agunt nisi soluta, unless they are in the same solution, which is somewhat already correct, but not quite. We now know, of course, that molecular interactions are the fundamental basis for basically almost all relevant processes, right? So with this in mind, what's ligand docking? At the core, ligand docking is, already the name suggests that there are two bodies, two entities that have to come together. At the core, what ligand docking is a computational technique that attempts to predict, all right, the three-dimensional structure of the complex, which will be formed between two molecules, usually a macromolecule. 95% of the times it's a protein, and a small organic molecule, usually. That's at least the way ligand docking is usually conceived. There are, of course, exceptions, and the field, like everything else, is moving forward. Then these techniques, these docking algorithms, provide a rough yet quick, yet fast estimate of the free energy binding associated to the formation of that complex. You already see in this slide, basically, a little bit of docking jargon, of lingo. I'm underlining that because I want to avoid any confusion. Sometimes the macromolecule, the protein, is referred to, and I will do that, and you'll see that in the literature. It is referred to as receptor, even though we are talking about a transporter or an enzyme. Yeah, in docking, we usually talk about receptor and ligand, which is usually, again, a small organic molecule, could be a peptide, could be in principle another protein. The technique, as suggested here, is somewhat rough. Doesn't mean to be exceedingly accurate, but this is a quotation I like very much. Our conquest, our grasp of reality usually starts with, idealizing with a simple model, which allows us to understand the complexity of reality. A little bit more practical, docking is usually a stepwise process. We could talk, for days about each one of these steps. There's abundant literature. As I said, most of you are already experts in this field. But again, just to give you an idea, you usually have to provide a representation of your pocket, which means at the basic level, you have to establish whether you know where your binding pocket is located, or you go blind. You make no assumption where your ligand is going to bind with respect to a protein. And then again, you have to provide a definition of this pocket in terms of which residues are involved in the definition of the pocket. What's their, for example, protonation state? Are there water molecules which will be relevant for the binding event? Are there ions, metal ions that must be considered part of the pocket, and so on. Then you have a posing phase, which is basically sampling the ligand conformational space within the binding pocket. And again, there are many different ways to do that. There are things you have to take into account, because usually a brute force enumeration of all possible degrees of freedom is not possible. So you need to play it smart. You need to employ algorithms that make this search efficient and possible in a reasonable amount of time. And last, as I already anticipated, you have a scoring phase. So to provide a quick, and you accept this trade-off between speed and accuracy, right? So it's fast, but not particularly accurate estimate of the free energy binding associated to the binding event, okay, to the formation of the complex you're representing. When you work with pockets and with systems which are at the interface between protein and membrane, even the very first step is not trivial. Sometimes you don't know exactly what pocket you're looking for. You don't know exactly the definition of this pocket. And this is where, an approach combining docking and molecular dynamics could become particularly helpful. The very first example, yes, of combining these two techniques is employing molecular dynamics as a pre-processing tool, which will help you to identify, well, at this stage, let's say, putative, hypothetical binding pockets. This is, for example, the work of Maria Ferraro. Some of you met her. She was involved in ALLODD in the early months, let's say. And basically, this is the core, the basis on which David's work is building upon. So you have probably heard already about this bit, and I'll keep it quite short. We were interested back then in looking into the serotonin transporter, which was known to basically have an interaction with cholesterol at the membrane level. And the idea, supported by experimental data, is that cholesterol wasn't just contributing to the physicochemical state of the membrane. It was actually interacting with the transporter in the way an allosteric modulator would. So we decided to study, let's say, this situation with the help of molecular dynamics to identify putative binding spots for cholesterol. And given that this task, couldn't be tackled especially, you know, a few years ago, by means of a fully atomistic simulation, at least with the resources we had in our hands, we resorted to a coarse-grained representation of the system. This is the serotonin transporter. And yeah, as I mentioned, we have experimental data suggesting that cholesterol plays a role as an allosteric modulator, not simply, you know, altering, let's say, the physical chemical state of the membrane, but interacting the way Ehrlich suggested, you know, tightly binding with, well, binding anyway, with the surface of the protein at the protein membrane interface. So that was the scientific question we asked. This is our representation. First thing, I'm going to go a little bit faster. Probably you already heard about this. We didn't have in our hands a model of the membrane, which would have allowed us for a detailed representation. So we ended up trying to create one. We know that we knew that this transporter partitioned preferentially in so-called lipid rafts. I understand this very concept is somewhat challenged by some authors, so we decided to investigate it anyway. So we came up with what it was experimentally reported to be a reasonable composition for the membrane. We tested several physicochemical properties. At this stage, we were only studying the membrane, right? And this was reported in here. I'm going to say it once. I tried, given, let's say, the lecturing tone of this seminar, to include as many references as possible. Some are from the work which is being presented. Some other are like what I believe to be classic work in the field. And, you know, when there are references to papers which are really seminal work in this field, I'll make sure to point it out to you. And as I said, so we analyzed our model of this raft-like membrane. We were convinced that it worked decently well, and this is the one we used for studying the interaction of cholesterol with the serotonin transporter. Again, this protocol is at the basis of David's work. It's a combination of a statistics-based approach. So we basically look into which residue preferentially interact with cholesterol across these very long coarse-grained MD simulations. We had multiple replica. We tested different concentrations of cholesterol, and we were basically looking for statistical outliers. So what you see here, even though it's quite squeezed, is actually a box plot. And what you see there are the outliers. So the ligands that statistically bind cholesterol. We also differentiated between two different situations. So where the cholesterol molecule comes and goes, and when it actually establishes a tight and continuous interaction with the membrane. At the same time, we used a density-based approach, standard implemented in the standard one implemented in GROMACS. And the overlap between these two approaches was considered a good proxy for hinting at the presence of an hotspot. So you can use this combination to identify pockets, or putative pockets at least, at the interface between protein and membrane. However, another very interesting way, in my opinion, and I'm going to spend a little bit more time on that, way to combine docking and molecular dynamics, especially again for pockets that you have just characterized, pockets where there's usually not that much structural information. You're lucky if you have one crystal structure or if you have some structural insights, and pockets which by definition, especially if you're looking for an allosteric situation, might be very flexible. And there aren't usually that many ligands. You see this, most of you are already familiar with the kind of situation I'm talking about. You could really use a protocol which takes into account receptor flexibility, and I'll explain in a second. So one question, especially those of you who are not modelers themselves, but get to work with modelers or interact with modelers, a question you might ask is that, okay, but this docking thing, does it work? I mean, can I trust the predictions that I get? Well, I can tell you that, first of all, already 20 years ago, so a long time ago, Golke and Klebe listed more than 20 drug discovery programs which brought a drug to the market, which benefit one way or another from, in that case, computational modeling in general. But out of those 20 programs, several were basically contributed by a docking exercise of some sort. Furthermore, when you perform a redocking exercise, what's a redocking exercise? You have a crystal structure of a complex, so an experimentally validated structure of a complex formed by, let's say, an enzyme and an inhibitor. You take the inhibitor out, you use your docking protocol, whatever it is, to dock this ligand inside the structure, and you compare the results with the crystal structure, with your ground truth, okay? In this case, when you redock, the accuracy is over 90%, meaning that you can generate the right pose somewhere, and it's between 80 and 90% after scoring, meaning that the best scoring pose is indeed the one more closely resembling the crystal structure, okay? However, is this a realistic situation? Not so much, right? And those of you, who are into these kind of things have already guessed where I'm going. Well, there are issues related to docking. I'm not going to discuss very much the first one already. Again, it would take quite some time. And furthermore, there are people way more expert than I am in this consortium to discuss this first bit. But scoring functions, as I said, are approximate, are geared to be fast, and most importantly, usually to discriminate binders from non-binders, are not particularly well suited to tell you, to rank binders, to tell you, well, if you introduce this modification in the structure, you're going to get a better compound. That's not what scoring functions for docking were originally developed to do. And for this reason, reasonably, they're not particularly good at doing that. There are different ways to do that. There are, computational methods which can really give you interesting results. So this is one problem, and I just wanted to mention that, but I'm not going into the details. The other problem is that proteins are flexible. I mean, the fact that most of the times in standard docking protocol, again, mostly for the sake of speed, we ignore this fact, is basically introducing a gross oversimplification in what we do. Proteins are flexible. They exist in a thermodynamic equilibrium between different conformations, which rapidly interconvert to one another, right? So a realistic docking protocol should, some would say must, take flexibility into account, right? So what happens if, remember my example from before, I have a crystal structure, I take the ligand out, and I dock this ligand back in. So the receptor is perfectly adapted already to lodge that ligand. What if this is not the case? What if I'm working, with an allosteric pocket I've just characterized with my coarse-grained molecular dynamics simulations? What if, I'm trying to dock a ligand which is completely different from the one with which the protein was co-crystallized? Well, in that case, we know, and we have known that for a very long time, and this isn't something I came up with, definitely, we know that the accuracy dropped to below 50%. So those numbers are not that great anymore, right? So you need to do something, okay? You need to find a smart way to take flexibility into account, okay? One way to do that, and this is one of the papers I was talking about. This is a review by Max Todorov and Ruben Abajan. It's very short, very brief paper, but a very nice reading. So you can use multiple receptor conformations. Okay, this is a very standard practice these days. We can define it as a discrete way to take flexibility into account. You pre-generate, either collecting experimental structures or, as we will see, using computational methods, multiple conformations of your receptor, and you use them for docking. Okay, so this is the protocol. You dock and score your ligand to all possible variants, and in this way, most likely, in selecting the best scoring pose, you have recapitulated, at least to a certain extent, the flexibility of the protein. I guess I can skip these. At this point, one of you might ask, okay, how many pocket variants should I have? Which one should I pick? I mean, how would I know in advance? Well, that's not, this is a very good question, and like many good questions, doesn't have a trivial answer. We tried to provide some guidelines back in the days with Manuel Rueda. The bottom line, this is going to sound trivial, but it actually works. When in doubt, you usually go for the larger pocket, the one with the greatest volume. Again, I know it sounds trivial, but it is actually going to help the quality of your results. What if you don't have multiple conformations of your receptors straight from crystal structures or cryo-EM or NMR or whatever? What if you only have one, and again, it's poorly characterized from a structural point of view? Well, this is where you can use a combination of molecular dynamics and docking. So you use molecular dynamics to sample the conformational space of that pocket, which you already have identified somehow. You generate multiple variants. You expect these to be small variants. I mean, I'm not saying, you know, you are going to explore radically different minima in the conformational space. We are talking about local variants of the same pocket, and you can use them for docking and for virtual screening. And again, it can be proven, and we did it basically in retrospective and prospective experiments, that this is going indeed to increase the quality of your results. Let me give you a couple of examples, okay? Again, probably shouldn't say that because I had the pleasure to co-author this one, even though, you know, I was basically tagging along. This is mostly the work of Marco De Vivo, who we have met in Strasbourg. This is another interesting reading, this review published in GEMET Chem on the role of molecular dynamics in drug discovery. And this is where this picture is taken from. Back in the days, we were studying the endocannabinoid system. So in particular, we were studying anandamide, which for those of you who don't know, is the endogenous agonist of CB1 and CB2. So one of the endogenous agonists of the cannabinoid receptors. In particular, we were interested in the way the signaling brought by anandamide is terminated. So the enzyme responsible for terminating the action of anandamide by degrading this neurotransmitter is the fatty acid amide hydrolase, which is a membrane enzyme. So it sits on top of the membrane thanks to this long transmembrane helix. Okay, so it's sitting on top of the membrane in the intracellular space. So what we knew is that anandamide, okay, is internalized, so it goes through the membrane, most likely by passive diffusion, because as you will see, that's quite a lipophilic molecule. Some authors suggest there might be transporters involved. Again, this isn't entirely clear. But what isn't clear is, you know, how the molecule then anandamide reaches the enzyme that actually degrades this transmitter. Okay, we came up with an interesting discovery, which is basically a splice variant of this enzyme called FLAT, as in Pha-like anandamide transporter. This is basically a catalytically inactive variant of the enzyme whose role is likely to shuttle anandamide toward the enzyme, right? And deliver anandamide to be degraded. Our goal was to find inhibitors of this transporter, the same way people have been looking for many years for Phi inhibitors, because if you artificially elevate the level of anandamide, you might obtain a therapeutic effect, say in neuropathic pain, which is what we were interested in. And other people have studied, you know, the role of anandamide, of Phi inhibitors in several other conditions. This is the bit about the splice variant. You see it lacks these alpha-1 and alpha-2 helices. So it's not anchored to the membrane anymore. And basically what is highlighted here, you see this red loop, which is the binding site. Again, it sits now at the interface between the protein and the membrane. And this alpha helix, which is usually there in Pha, is not there in FLAT. So this requires some molecular dynamics to model this situation, because you see, when you take the alpha helix away, it changes basically the electrostatic environment, which in turn affects the catalytic machinery. And then you have this loop here. You see that most likely changes conformation. So you have a rearrangement of part of the binding pocket of putative inhibitors, well, at the interface between the protein and the membrane. And most likely you have a different role for water. So you have basically you have more water given the lack of the structure in front of the binding site. Okay. So with this in mind, we indeed proceeded to model through molecular dynamics this protein, and we started the virtual screening. So we were looking for these flat inhibitors. What we knew, we started with this MoldCard. Back then I was using MoldSoft ICM. We used MoldCard, and we didn't want to, you know, screen blindly this entire collection. We made an assumption. I mean, we look at the binders. This is anandamide, so that's the substrate, let's say the transported molecule. This is AM404, which is a known transport inhibitor. So there were transport inhibitors before the transporter was actually discovered. Again, AM404, 123, OL92. You see, these are very lipophilic molecules. So we had a pretty good idea of the kind of physicochemical profile we were looking for. So we came up quite empirically, let's say, with a set of filters, which we applied to the library. We did the docking, but before doing that, we weren't really interested in molecules which were trivially similar to those we already knew. So those molecules, very close in terms of Tanimoto distance to non-binders, were discarded. There you go. These are examples of molecules which were basically discarded. We ended up with over 100,000 structures, which we docked, implementing flexibility. So we had multiple variants of the pocket thanks to our molecular dynamic studies, and we took that into account in our screening protocol. In particular, what we use is a technique which I contributed to develop and which is implemented in ICM, which is called 4D docking, and if in case you're interested, there's the reference over here. So it is based on that idea I was mentioning before of multiple receptor conformations, and it's applied to docking and in this case to virtual screening. In particular, you see the role of flexibility is mainly related not just to that loop I was mentioning before, but to the side chain of this phenylalanine. You see that, you know, can exist in different conformations. Okay, so the shape of the binding pocket is this sort of Y. And one of these two channels can be alternatively occupied based on the conformation of the phenylalanine side chain. And this changes, of course, the ranking of your ligands if you consider only one variant or the other quite substantially. So we used the 4D docking. We looked into the binding score. We did some cluster analysis, I mean, chemically based cluster analysis. We ended up with 46 compounds after visual inspection, which we tested. One of them turned out to be fairly good molecule, which ARN272, with this phthalazine over here. So you see it binds the transporter, so it's active in vitro studies. We can prove that it blocks the accumulation of anandamide, so it's active in cellular study, transport studies, and most importantly, it's active in an in vivo model of neuropathic pain. So this was tested in rats, okay? And you can see the dose-dependent activity of this compound. This study was published and the molecules were patented. Another example of combining flexibility in virtual screening in pockets which are somewhere at the interface between the protein and the membrane comes from my interest in polypharmacology. In particular, my long-standing interest is in nicotine addiction. So this is the bit where I try to scare you about, you know, not to smoke or quit if you're smoking. I'm not going to, I mean, given this audience, given that the people here are possibly already quite informed about the danger of tobacco addiction, I'm not going to spend long on this topic. Just let me mention, I just want to spend 30 seconds saying that the idea that nicotine addiction is a sole problem is not true. I mean, the prevalence in the population in Europe is fairly high, and in developing countries is very high. And most importantly, in several fractions, several groups in the population, chiefly people affected by mental health disorders, this prevalence can be as high as 80%. So this isn't just an healthcare problem, this is also an inequality problem, okay? And nicotine addiction is quite complex. As you know, it's caused by this alkaloid here, nicotine, which per se, it's fairly innocuous. Nicotine is what makes you addicted to tobacco, but you know, what will eventually give you cancer and kill you, because that's what's going to happen. I mean, it's just a matter of when, it's not a matter of if, are the burned substances in tobacco smoke. This addiction is very strong, and it basically affects most of all relevant circuits in the brain. This is why we thought this could be a condition that possibly could be tackled with a multi-target approach. Jen Carlson gave a very nice introduction in Strasbourg to this topic, so I guess I don't have much that I can add to what he said. In particular, in this case, we were targeting one traditional target for nicotine addiction, which is the dopamine receptor subtype D3, which is a traditional target in nicotine addiction. And again, this enzyme that I've already mentioned, which is the fatty acid amide hydrolase. In particular, though, first we were interested in anandamide. This enzyme's pharmacology is, you know, quite promiscuous, so to speak. It also degrades these other molecules, olehyl ethanolamide and palmitoyl ethanolamide, which are in turn PPAR alpha agonists. So this is the part we are interested in. If we keep artificially high, we elevate the level of these two molecules, you know, we obtain a stimulation at PPAR alpha that through this pathway, again, is going to interfere with the mechanisms of nicotine addiction. In particular, the idea was to interfere with relapse. Okay, not that much with cravings and promoting quitting, but avoiding relapse. This was a very, very chemically driven project at the beginning. These are your prototypical Pha inhibitors, the fatty acid amide hydrolase inhibitor, URB 597. Interestingly, URB 597 stands for Urbino, so I guess that was destiny. And this is BP 897, a prototypical D3 antagonist, okay? We thought, well, these are two pharmacophores that we can indeed combine together through these carbamates. And this is exactly what we tried to do. In principle, there are many, many molecules you can do. So we came up with an in silico, so this is just inside the computer, in silico library of compounds, combining together different moieties and groups traditionally associated either to D3 or Pha. We docked each one of these compounds at the two structures. We got lucky back then because we already had, even though those were relatively early days for GPCR crystal structures, we already had a nice crystal structure of the D3 receptor, and we already had the fatty acid amide hydrolase crystal structure, even though back then we only had the rat one, not the human one. Anyway, this is the binding, the predicted binding mode at D3. In particular, what's interesting for us is this bit over here, which is the so-called specificity pocket. You have this pocket in D3, you don't have it in other subtypes like D2. Okay, sometimes, just given that this is ALLODD, sometimes in literature, you erroneously find this pocket referred to as an allosteric pocket, which is not. This is the orthosteric pocket. You see, this is conserved position 332. The interesting bit is that here you basically already have the membrane. So you are already at the interface between TM7, TM1, and the membrane. Okay, so you need a little bit of modeling in this region. Same for Pha, which I was mentioning before. This is the channel that, you know, and these two channels are basically connected to the membrane. Here you would have that phenylalanine I was talking about before. Please note that in case of the fatty acid amide hydrolase, we are talking about covalent inhibitors. So here we have the added layer of complexity of studying basically covalent inhibitors. And we docked all these compounds. we had some level of confidence in this binding mode thanks to previous QMMM studies carried out by my colleague Alessio. And what we did eventually, thanks to this docking study, we prioritized two compounds, 654 and 636, where the compounds we synthesized, tested. Please note that there is no cross-activity between D3 modulators, no activity at Fa inhibitors, no activity at D3. Our molecules were single-digit nanomolar at both targets. So this encouraged us quite a lot. We worked a lot on this series of compounds. This isn't the topic of the talk today. For those of you who are curious, I collected some references. We studied the solubility, which those of you have, the MedChem I already eyeballed the lack of solubility of those compounds. We ended up synthesizing more than 60 molecules. We had an activity at CB1 that we couldn't explain. We eventually were able to model it and design it out. Again, we ended up with this one, which is our front runner, ARN153A1. It's fairly stable. We proved that it's brain penetrant. And very recently, in 2022, we eventually obtained this very nice data, see that at a reasonable concentration of nicotine used for pre-training, precondition the rats, we can see clearly that we have an effect in a behavioral study counteracting nicotine addiction. So that was pretty cool. Then I'm going to move to another topic, which is a little bit more, let's say, intuitive. And people have been doing that for a very long time. You can basically combine docking and molecular dynamics to test the stability of your pose. Okay, so I mentioned before how we indeed, you see, we did that with this is ARN272 at the binding site of FLAT, you remember the transport inhibitors. And what we did was basically, like, I know this sounds a little bit naive these days, right? So we have 10 nanoseconds stability test. You see that in orange you have the pocket backbone. We said, well, the molecule is fairly stable. We have the enzyme, the membrane, water. So quite a huge system. That's why we couldn't afford back then a much longer simulation. Not that we tried multiple times, multiple replica, and the pose was fairly stable. So you see the point. I mentioned how scoring functions cannot be entirely trusted. And I also mentioned the fact that proteins, we usually don't study flexibility in standard docking. So you end up with a pose and you want to increase your confidence in that binding mode. That's why this complex that you obtain can undergo a molecular dynamic simulation, okay? This is basically the idea. So a stable pose will tend to display a very stable RMSD, doesn't move much, basically, whereas an unstable one, an artifact, will fly away. Furthermore, you can study local induced fit effects. You can study the role of water, which again is usually not explicitly taken into account in your standard docking simulation. You only have a very low dielectric constant inside the pocket, right? And this is what people have done since forever, including us. This is a paper from when I was a very young PhD student, and we studied the stability of poses of propedium at acetylcholinesterase. Okay, so we were interested in Alzheimer's. In particular, I was studying the stability of ligands at the protein membrane interface. That this is a system I think Sonja mentioned more than once. This is the C5a receptor, and this is published work from when I was working in the ComChem team at Sosei Heptares, which was Heptares Therapeutics back then. And we published the crystal structure of this receptor, C5a, which you see here as a dimer. Whether it actually exists biological as a dimer or not is not entirely clear. And again, this isn't something I'm going to discuss right now. The interesting bit is that we have this ligand, you see that at the protein ligand interface, NDT, And we wanted to understand the role of this allosteric modulator. And in order to do that, we resorted again to molecular dynamics, starting, well, in this case, at least for this ligand, from a crystal structure, not exactly from a docking pose. And what you can see that is when, sorry, this one, yeah, apologies. So if you take the wild type, the wild type receptor, the ligand, I'm gonna go with a little spoiler over here, stays there. It makes this stable interaction with that tryptophan residue at the, again, we are here, I'm not showing the membrane for clarity, but the ligand is basically surrounded by the membrane, right? We are at the protein membrane interface. Whereas, if we introduce this mutant, so we convert the tryptophan into a leucine, you can see that in a very short amount of time, the ligand flies away. Now, the interesting part is that for many years, it was believed that this ligand, NDT, would bind at the orthosteric binding site. And that this data from mutagenesis was known. So the result was that people were trying to model the protein with the tryptophan inside the orthosteric pocket, which in turn would basically compromise the quality of the overall model. This is to say that, again, lesson learned here, I guess, the Occam's razor not always works when it comes to biological data. The logical explanation, the so-called simplest one, would have been this tryptophan, since we know from mutagenesis that if you change it, you compromise the efficacy of this allosteric, of this modulator, well, it should sit inside the binding pocket, and that was not the case. You see, in just a few nanoseconds, we had multiple replica, we tried different things, but the message stays. In just a few nanoseconds, the ligand flies away. We also use molecular dynamics to explain the mechanism of action. Basically, this ligand is like a wedge, okay? It inserts itself among TM3, 4, and 5, and makes it impossible for these helices to change their conformation. So in this way, basically, it exerts its allosteric modulation. And interestingly, when you remove the ligand from this picture, almost immediately, again, with very short molecular dynamic simulation, you can recapitulate what's happening to the pocket. As expected, somehow, the side chain of the tryptophan flips and occupies the empty space, which would have been otherwise occupied by the modulator, okay? So people have used this combination of I dock and then I run molecular dynamics to test stability for a long time. There are, let's say, computational modeling software that implements this strategy right away. We use that. In particular, I want to suggest, I'm not involved with these two papers, so you know, I just want to mention that I found them very interesting reading. These authors, they work for Takeda. They basically studied once and for all in a systematic way, so with a very huge benchmark and using redocking, cross-docking, running multiple replicas. Furthermore, they used glide and then Desmond, so avoiding the problem of shifting from one force field to another. They systematically studied this combination of docking and molecular dynamics. And I really encourage you, if you're curious, to read these two papers. Very last thing I want to mention is that docking alone can somehow be considered the computational counterpart of a binding assay. So it's not going to tell you how the ligand goes in, how the ligand goes out. You're not going to get from docking kinetic data. Binding kinetic, computational predictions of binding kinetics is another thing that interests me quite a lot, and maybe we can talk about it another time. But we thought, well, maybe a combination of docking and molecular dynamics can instead be considered like the computational counterpart of functional assay. So we can predict efficacy. In particular, if we go back to those carbamates, which we were studying for nicotine addiction, we noticed something that is fairly common in GPCRs. This has been reported. I thought I would have had a very nice perspective from a few years ago in JMed Chem. I apologize for that. For those of you who are interested, I can dig out the references and provide it to you. This is known in GPCRs. Very small changes in the structure of, let's say, a modulator can shift quite abruptly the efficacy profile from, like in this case, a molecule which is a neutral antagonist to a molecule that, you see with just a few little changes within the experimental error is basically as active as dopamine. Okay, very small modifications. We set out to understand whether we could make some kind of, we could rationalize that at least a little bit, and ideally even try to make some predictions. This was a collaboration with Vicky. Sonja already mentioned that I was involved with founding Vicky, so I guess my possible conflict of interest has already been disclosed. This is the protocol that we used. Again, this was quite a few years ago, but we had the dopamine D3 receptor in membrane and explicit water. We basically docked our antagonist, our partial agonist, and what we call the full agonist, 90% efficacy agonist within the binding site. So we treated each system independently. We had multiple replica, 3 microseconds each, which was a reasonable, amount of simulated time, especially at the time these simulations were carried out. As you see, this is again Maria Ferraro's work when we were working together. And then we basically performed some clustering. So this is the output of a cluster analysis. This is not a Markov state model. I want to mention that very clearly. And here we basically look at the way, if you combine an agonist, an antagonist, and a partial agonist, and you run cluster analysis using both determinants from the binding site and what the ligands have in common, you can see that different ligands partition in different clusters, but not exclusively. That's the point. Because otherwise you might say, yeah, well, good work. Your cluster analysis basically separates 3 independent trajectories. That's not what happened. I mean, you have that certain conformations are preferentially explored by an agonist, certain conformations are preferentially explored by an antagonist, but not exclusively. They explore them, but with different frequency. And if you look into this kind of plots, basically you can identify which conformations belong preferentially to a certain type of efficacy, something that docking alone would not show you. So you see that, for example, this is an example from one of our molecules. You see how subtle changes can be explored within the binding site, shifting from antagonist and agonist and so on. Again, I guess I'm almost at the end of the time I have available, but I want to mention that we challenged our model a little bit. Again, I don't want to overstate the importance of that. Those of you, again, working in chemistry would be quick to stress that, well, given the SAR you just showed us, I could have told you that that's a partial agonist. You don't need all these simulations. So this isn't exactly a prospective experiment at this stage. It's more like validating the protocol once again. And sure enough, this compound displays the typical features of a partial agonist. And you can very much distinguish from an antagonist and a partial agonist. I'm not claiming we see any activation of the receptor, okay? So all docking poses were obtained in combination with the crystal structure of D3 we had available, which is the one with the thiocloprid, so it's an antagonist. It's an inactive state conformation of D3. Conclusions. I think that we are moving toward a unified framework between docking and molecular dynamics. Some authors have tentatively introduced the term dynamic docking. I don't think we are there yet. There are very interesting studies going on, but at this stage, we are still bound to, let's say, combine two independent techniques. There isn't this unified framework. I don't feel like saying it's already there yet to be routinely employed, on a daily basis in drug discovery. Eventually, I mean, the obstacles, which are the usual, we need better software, better hardware, will lead us to overcome these, very much like this definition by Carlson and McCammon, this unfortunate but necessary trade-off between speed and accuracy. Okay, and at this stage, what we can do is to combine using docking with molecular dynamics, using the latter as a pre-processing, post-processing tool, and especially when we are dealing with pockets like those of the protein membrane interface, like allosteric pockets that are flexible by definition. There isn't that much information in terms of known ligands. So even a chemogenomic approach is not delivering that much in this case, and you don't have that much structural information. Okay, so I guess that's the end of my presentation. I thank you very much for your attention, and I encourage you to ask questions if you want to. Thank you.
Thank you very much for this wonderful overview and comprehensive introduction also in how we can apply MD for post-processing and also as a potential functional assay in drug discovery. So yes, I would like to open the floor now for any questions, if you have. Maybe I can start with one. You showed really nicely how you use mutational data for C5a complement receptor. Can you give some systematic advice on how we can use experimental data such as mutagenesis studies or also some NMR data that we can integrate them in some of the computational approaches, especially, yes, I think also in a membrane environment?
Yeah, I mean, it's always hard to give, insightful general advice on something which is very, well, I guess the general advice is, at the cost of sounding a little bit paradoxical, is that it's very system dependent, right? Mm. I think that if you're interested in something which is quick, which should give you some insight, some actionable knowledge with a relatively short turnaround, you really need to make sure that, let's say, example, your mutation directly affects, let's say, binding. So there's an hydrogen bond which is missing, or there's a salt bridge which cannot be formed. Whereas if your mutation has a more subtle effect, such as it promotes a slight change in the conformation of your protein or something like that, then you need, something which necessarily takes longer. You need, for example, a longer simulation in order for your protein, for your target, let's say, to adapt, starting, I assume, from, what is indeed the structure of a wild type, and you artificially introduce this mutation in order to possibly explain and to explain the experimental data and use that for, in turn, maybe develop new, better molecules. Right. So, yeah, sometimes you really need to, to improve, let's say, the amount of computational time that you are willing to put.
So I see Marco.
Yes, hi Giovanni. Interesting talk. So I would like so when you do a prospective drug discovery study and you do your docking or you do your computational studies to identify potential hits, and then you have to experimentally test them to verify and validate your predictions. So how many compounds do you think you need to test if you don't know the answer in advance? And the second thing, so to find a hit. And the second question is, what is actually your definition of a hit?
Right. Okay. Can I start with the second one?
Yeah, sure.
Again, it's very much system dependent, but on average, let's say my definition of hit is definitely something that shows activity in the sort of assay which I'm going to use for later on for development. It sounds trivial, but sometimes you get a hit from some biophysical technique, and then you know that for development, for whatever reason, you're then going to use a binding assay with purified enzymes in a biochemical setting. So ideally, I'd like to be consistent. I usually, for your typical target, I look into something which is already in the low micromolar range. Say, I don't know, if you want a threshold is somewhere between 50 and 10 micromolar on average, okay? And what I look for is chemical accessibility. I mean, I try to look at the hits with the help of an actual medchem, and if at all possible, because of course, sometimes you don't have the luxury to be picky, but if at all possible, I'd like to start working with a structure that my colleagues in the synthetic chemistry lab would be happy to work with. Mm. This is for my definition of hit. I tend to, I don't know, my feeling is that sometimes people get filter happy, as you know, in trigger happy, but with filters, and you end up with nothing, or with just molecules, I don't know, which are not very diverse, chemically speaking. So a preliminary hit, even if you have some groups which you don't particularly like, there will be time during development to get rid of those. And solubility is definitely something again, it can be improved, but most of the time, some of these molecules are like useless because you expect, again, an activity which is in the low micromolar range, and sometimes the solubility at that range is already an issue. And instead, basically, so your first question, how many, as much as I can afford, that would be the straightforward answer, which could be afford, meaning depending on my budget and depending on what my partners doing the actual screening are willing to commit in terms of time and resources. I was working with a colleague in Birmingham, okay, on a completely different project. And these molecules would be tested on primary cultures, straight from tumors. So he said, I asked pretty much a variant of your question. So how many molecules do you want? And he said, well, in the next 6 months, I guess I can test 2. And I'm like, okay, then, you know, I really need to build some confidence on the molecules that I'm going to suggest. That was probably not enough. I don't know what would be the lower limit, but 1 or 2 molecules, I have to question whether if it even makes sense, you know, to narrow it, to create those funnels from like 2 billions, which is the number you expect these days from ultra large libraries, and narrow it down to like 2 compounds.
But yeah, so to clarify a little bit, because my question is how many compounds you need to test in order to reduce the risk to an affordable level, because that's also another problem, right? So you invest time and you invest resources to screen, and then you want to see results. And if you don't see, that's, I mean, generally the project stops. Or well, so my simple question is, would you like to test 10, 100, or 1,000? So what is your order of magnitude in order to be successful?
In my experience, hardly above 100. I could hardly afford to test compounds above 100. I think one time, this is a published paper in 2021, those were inhibitors, collaboration with the Italian Institute of Technology. The first author is this guy by the name of Di Fruscia. Anyway, we were able to perform a mid-throughput screening, and we ended up testing a couple of thousand compounds. But at least for me, that was a one-off. I usually can afford that much in an academic setting. 40 compounds, 50 compounds, a little bit more if I get lucky. It's also an exercise of expectation management. Sometimes people expect, that you test 10 compounds and 1 or 2 are going to be active, which is great if it happens, but that is not usually the case. You learn from the first iteration. You remove those compounds from your batch, and you maybe have to test 2 or 3 times before finding a hit. Also because, you find a hit, but then, you find a hit, you find a molecule that displays some activity, and then you want to verify whether you can work on that molecule. Sometimes, for whatever reason, the molecule works in a preliminary assay, but then whatever modification you introduce, the activity drops. And so that's, not actionable from the chemical point of view. And so.
Okay, thank you very much.
Thank you, Marco, for this question. Anyone else wanting to pick and get some inputs? Now is your time. So I think yes. Oh yeah, Ravina, please go ahead, unmute.
Do you hear me?
Yep.
Do you hear me?
Yes, we do.
Okay. I have a question regarding the using MD simulations for like the checking the pose, the binding pose, the docking pose stability, like the post-processing MD. Have you encountered like again false positives from this kind of post-filtering? If like even with MD, the suggested pose is stable, but actually it will not actually happen if the ligand is outside, and then in order to go inside, it's not possible, the binding. Like, have you encountered this kind of false positives?
Yeah, if I understand correctly, the point is you dock your ligand, you get a decent score or a good score, then you run ideally a few replica of not so short MD trajectory, and the ligand seems, the ligand is stable across the simulation. You test the compound and the compound is inactive. That's the situation you are describing. So despite the fact that you also use molecular dynamics, you still bump into a false positive. Yes, it did happen, but I think that we should look into the combination of docking and MD as a confidence building exercise, which is gonna be completed. So it will reach the stage where, yes, I'm positive that this molecule works only when you get to test it experimentally, possibly more than once, from different operators and whatnot. This is just, bringing you a little bit closer to that, let's say, level of confidence. Again, at least in my case, it usually involves either investing some money or someone else's time in purchasing or synthesizing that specific compound and have it tested. So I feel like, I want to invest a little bit more, provide a little bit more confidence before suggesting that is the molecule or one of the molecules that we should indeed test at the end.
Thank you. Thank you for the answer.
Thank you, Ravina, for your question. Any last question? Otherwise, please, thank you again for this wonderful seminar, and I wish you all a wonderful afternoon and goodbye.
Bye, everyone.
Thank you very much.
Take care.
Bye.
Bye-bye.
Thank you for the
Bye.
seminar.
Bye-bye.
Thank you. Bye.